All right, so today we will be talking about cloning technology. So, cloning technology is producing identical copies of DNAs, cells, or organisms. In biotech, this typically refers to gene cloning, not reproductive cloning. So, not making a copy of your dog or yourself. We're talking about genes specifically. So, there are three different types of cloning. We have gene cloning, cell cloning, and organism cloning. Gene cloning, the purpose is to make copies of a specific gene. A gene is inserted into a plasmid vector. That vector becomes a recombinant plasmid and is inserted into a host cell. Typically, this host cell is E. coli. Um, as the cell replicates, it clones the gene, producing many identical copies. So, why is this super important? Well, we've already kind of talked about recombinant DNA and recombinant genes. It's important so it enables the production of proteins like insulin, human growth hormones, things that we need to produce medication and help in research and allow gene studies or therapies. There are 6 steps in gene cloning. We first isolate the gene we're interested in, then we cut it with restriction enzymes. We insert that gene into a plasmid vector, forming a recombinant DNA. Then we transform the plasmid into the host cell, often a bacteria. So we put the plasmid into the host and then select for transformed cells, usually via antibiotic resistance or things like that. And then we allow the host cells to replicate, cloning the gene. Cell cloning is to create identical copies of a single cell. How this works is a single cell is allowed to divide and grow into a colony of genetically identical cells. Often, this is used with stem cells or immune cells, and it is critical in research, cancer studies, drug testing, and therapeutic applications. And then we have organism cloning, and this is to produce a genetically identical organism. The first organism to be genetically cloned was Dolly the sheep. Uh, it involves stomatic cell nucleuses being transferred into a embryonic cell. The nucleus of a stomatic cell is transferred to an enucleated egg cell. So an egg cell without a nucleus. The egg cell is then stimulated to divide and develop into an embryo. A great example of this, like I've already mentioned, was Dolly the sheep. She was the first cloned mammal from an adult cell. Why does it matter? It's not super common in biotech labs, but we can use it for endangered species, conservation, research, and agriculture. So we've seen that really similarly now with the dire wolves and the woolly mice. And that is all for this section of lecture. I will see you guys next time. Bye.